sea looks blue. Now, what most of you would think is that this is because of the reflection of the sky. And in fact, I always say that it is never wrong to think in that way, but we'll try to find out if really the sky, reflection of the sky is the reason why the water looks blue. Now I take another photograph, and these photographs are not mine. It is there in the internet, and I've actually just picked an. You would have taken many photographs, and you would have found out that in waterfall also you see a coloration of the water, and this water color is not blue, but it is green, and many would think that it is because of algae or blue-green algae or something, or it could be the reflection of the vegetation around that gives you this color. But if suppose it was blue-green algae, all of you are biologists, so you know that a flowing water generally is supposed to be highly pure. So this is a flowing water, and so we should not have any algae growing in this water. So that puts to rest that argument. And the other argument is a reflection of the vegetation around it, and that gives you the color. So I've taken another picture, again from the internet. Here is a pond or a pool which has been created in front of somebody's house, and the water looks blue. And the vegetations are there, and the sky is there. So what is the reason for this color? But I want you to concentrate on this region. This is the shallower end of the water, and you can see the bluishness is not there. It looks, in fact, transparent, more or less transparent. You can see that there is no coloration to the water. So that means the water cannot be having a reflection, because if it was reflection, then it should be having in this place also. And here also we have a pond which is not very deep. So it is not like the sea, but still it gives you color. Now this is what puzzled C.V. Raman, and in fact C.V. Raman started investigating this, and that is, I'm going to go forward with that argument. But what exactly is happening here? So water, though is transparent, it actually starts absorbing colors of the light. That means different wavelengths of light, when it passes through different distances of water thickness, it actually gets absorbed. This is there for the red first, then orange, yellow, then green, blue, ultraviolet, violet, and indigo and violet. All these colors get absorbed, and that is the reason when you actually see a National Geography movie of a deep sea marine life, you would find that they have to use, in fact, torchlights to see the living organisms inside a deep sea. All the water, the thickness of the water is very, very important in removing off the different colors of the visible light. So in fact, when shallowness of the water is there, you would find that it still is transparent. But as the thickness increases, you will find first the red, blue, and green, which would red, yellow, and orange, which gets absorbed. That is what gives you the greenish color, because our eyes are very sensitive to green. And once the green is also absorbed, you will get the blue color. And this blue color gets reflected back because of the, something called total internal reflection. And that is the reason why you actually see the blue color. So in fact, Raman did this experiment, and he did publish a Nature paper in 1921, much before his discovery of Raman, the color of sea. And here I want to highlight the fact that all of you, if you open up your 12th standard textbook or look at it, you know that light, when it is reflected, is partially polarized. If the light is unpolarized, it gets partially polarized. So if you put in a polarizer, you would actually be able to see decrease in the color. In fact, this is the technique used when you take a photograph in a bright sun day or sunny day. So what you do is you put polarizers in front of your lens to cut off all the reflections. So you can actually cut off the reflections if it was only reflections. So what he did is he used to carry always nickel prism with him. So he started looking at the sea and started observing the sea very carefully. And when he observed with a polarizer, he found that the light never gets cut off. In fact, when it gets richer. So this is what he has actually told. Observing tolerably smooth patch of water with this held in front of the eye at approximately the polarizing angle with the surface of the sea, the reflection of the sky may quench by suitable orientation of the nickel prism. So he did this experiment. In fact, he was going in the ship and he did this experiment while going and coming back, and he wrote this and he published it. He was in a hurry. He published it from Bombay Harbor itself. He didn't even want to go to his lab or anywhere else and wrote this paper finishing this experiment. That is Raman. So he said this has to be something which is very, very important, that you are actually seeing something from the light. So this is the interesting aspect. Now, another aspect is that most of us know that light interacts with matter, and the things which happen is transmission, reflection, then absorption, and you also have luminescence. These are things majorly happening, and many of you may say diffraction and interference. In fact, diffraction and interference is actually a result of transmission and reflection and absorption and luminescence, but it is actually coming into your eyes, and that in the eyes, this light is actually interfering. So it is not exactly in the material, but from the material, the light actually gets transmitted or reflected, and that actually comes into your eye and does it. So that's why I never. Now, the other thing which happens, and that is very, very important, I think there is some echo. Somebody has switched on the mic or something. Can everybody mute? Yeah, sorry. Thank you. So basically, elastic scattering. And whenever you say elastic scattering, everybody knows collision. So if you take a ball and throw it onto a wall, and any tennis ball or any such good ball, it would come back with the same energy you threw it. And that's what is called elastic collision. So when you talk about elastic, it means it actually has no change in energy. So in light parlance, it means energy doesn't change, and energy is h nu. So nu frequency doesn't change. That means the same color. So elastic scattering is light coming back to you by interacting with the molecules and atoms inside the material and scattering, coming in all directions. It is different from reflection, because reflection follows the Snell's law, which is angle of incidence is equal to angle of reflection. All this has been taught to you from 12th standard. Now, when I always ask this question when my audience is there, that which of these phenomena is the reason why I see you? And most often people would say reflection. This is what you have always thought, that it is because of the reflection. But if I actually give you the examples where you would find it difficult to explain that reflection is the reason. All of you know that good reflection comes only if the surface is smooth. And what happens when you go to take a photograph in a studio? They have kept not anything but powder. When you have powder, if you put powder on your face, it doesn't give you good reflection. It gives you a matte finish. It doesn't give you a glossy finish. So that itself says that you don't want good reflection. That is why you do. And the second observation you have when you take a photograph inside a studio, the light is not put on to you straight on your face, but it is actually an umbrella. White umbrella is put in front of the light to diffuse the light. That means you want no direct light to fall on you. And that means the reflection is not the principle behind it. It is because of the elastic scattering. So in fact, elastic scattering, it helps you in seeing each other. And this is the reason when you turn off all the lights in the room, still I can see you if a faint light gets inside the room. And that's the beauty of elastic scattering. Now, along with that, there is something called the inelastic scattering, just like the inelastic collision, where the energy changes. And whenever the energy changes in light, the color of the light changes. I have only accentuated the light color. I am saying that it is blue or red, but it is not blue or red. It means blue shifted or red shifted. That is the meaning of these colors. So in fact, this is what Raman actually proposed, that in the scattering of light from the medium, there are inelastic scattering of light, which actually gives you information about the material. And this is the discovery which he got him the Nobel Prize. Now, to show you that indeed there is inelastic scattering of light happens, I am actually showing you an example taking cyclohexane. All of you know cyclohexane is a transparent liquid, and I have put it inside a bottle, and I am putting a blue light. And you can see if you didn't have this, the light would travel without any change in its direction, because lasers are completely confined into the volume of light which goes in the forward direction. So if I am taking a photograph of this at 90 degrees to it, whatever is coming is elastic scattering. Now most of you would be confused, why does it look white in color? But all of you know that if I take a photograph when there is bright light, then I would saturate my detector, and my detector cannot distinguish colors. And this is what you have seen when you take a photograph in a bright sunlight. You can't see colors very well because the brightness of the light is too much that it saturates your lens. Nowadays, cameras come with that adjustment of the pupil so that you actually can not saturate the detector. But here the light intensity is so much that it is actually giving you a white color. You can see the entry point of the laser light itself is white in color. So it is not white, it is the blue light. If you will actually look at it carefully, it will be completely blue light which is coming out. This is elastic scattering. Now, if I want to see inelastic scattering of light, I have to hide this light. And this is done by filters. And you have very good filters which filter out a specific wavelength of light. So I have put a filter, and you can see the elastic scattering of dust particle is gone. You can't see the elastic scattering of the dust particle, and you can see now the color is different from what the color you saw. Again, here whitish color comes because of the intensity of the light. So you can see this is the Raman scattering of light. And in fact, this is clear evidence of seeing Raman with your naked eyes. And in fact, Raman could discover this only because of this reason, that this inelastic scattering of light is intense enough from organic molecules that you can actually get this done. Now, in general, we need to always look at what is this shifted frequency. That means how much is red shifted or blue shifted. And this shifted frequency is in energy is equal to the energy of the vibrations of the molecule. And in fact, it is not only vibrations. All of you know that vibrations are coupled with rotations, and vibration is coupled with electronic states. So you can actually have rotational Raman, vibrational Raman, electronic Raman. All three Ramans are possible, but we would not be concentrating on these two, but we will be concentrating on vibrational Raman only. So now you can see that when I am actually looking at the vibrations of the molecule, if any diatomic molecule is there, it actually vibrates very, very uniform way. That is only it will stretch around the bond, and this is how a vibration of a diatomic molecule will take place, and this is the only vibration present in the diatomic molecule. And what you can see is that this molecule is HF and this molecule is HCl, and the difference between this is there is a chlorine atom which is heavier than the fluorine atom. And because of which you can see this is vigorously vibrating while this is vibrating much slower. And that means it is less energetic compared to this one. So that is how we write the energy in terms of centimeter inverse. But centimeter inverse usually is a length scale. But if you multiply with Planck's constant and speed of light, then this becomes EV, electron volts, and that is unit of energy. And that is roughly 8,000 electron volts. A centimeter inverse is 1 electron volt. So this is roughly 500 milli electron volts, and this is around 300 milli electron volts. So you can see that mass has an effect on the vibration, and the bond between them has an effect on the vibrations. Now I have just added one more atom. The moment I add one more atom, the number of vibrations increase, and you can see there is symmetric vibration, asymmetric vibration, symmetric bending. And in bending, you have bond angle change, and in stretching, you have bond length change. And all of you know that bonds are very difficult to move around, and hence this is more energetic compared to this, and that's the reason the energy of the vibrations is less compared to the stretching. Now I have added one more atom, and I see an increase in number of vibrations, but the number of vibrations do not increase exponentially. They increase in a very particular way, and you can see that there is stretching, asymmetric stretching, then you have bending, symmetric, and asymmetric bending. So these are the types of vibrations which molecule would execute, and if I increase the number of vibrations, sometimes it is possible to get many vibrations, and sometimes it is found that you have very few vibrations. Here in this case, your molecule is an octahedral molecule, is SF6, 7 atoms in it, and you can see there is symmetric stretching, asymmetric stretching, and asymmetric stretching again, symmetric bending, asymmetric bending, and asymmetric bending. So you can see when it is bending, it is only the bond angle change. When it is stretching, its bond distance changes. So you can easily see that there is these things happen, and energetically they are different. But if my symmetry of the molecule changes, the number of vibrations would increase because there are some degeneracy in the vibrations. And that is what is immediately telling you the molecular configuration. If there is a symmetric molecule, you will have a very clean Raman spectra. Whereas when you have an asymmetric molecule, you will have many Raman modes coming out of it. So this is a clear indication of whether your molecule is symmetric or not symmetric. Now, very importantly, all of you in your 12th standard would have studied something called simple harmonic oscillators, and its frequency is given by K by reduced mass, and K is nothing but spring constant. This is what has been taught to you in 12th standard if you have gone back and looked at it, or some of you may be parents and you would be having 12th standard child and he would be studying this. So this is basically what happens. Now if you consider that this is not a two ball connected by string, but this is two atoms connected by a bond, then it also produces a simple harmonic oscillator where this is the bond stiffness and this is the reduced mass of the atoms. Now, because it is a molecule, you would have charges associated with it, and because of that, nucleus is positively charged and the surrounding is electrons negatively charged. Now, whenever there is a charge, in principle, the molecule is charged neutral, and because it is charged neutral, you would not have any effect of, you won't see the effect of the charge. But if I put my electromagnetic radiation, it has an electric field. And all of you know electromagnetic radiation is a sinusoidal or a cos functional variation. The electric field moves in a wavy pattern, and it produces magnetic field which is perpendicular to it, and it is also wavy. Electric field, whenever falls on the molecule, it will actually separate the charges. it will produce an dipole instantaneously, and since it is oscillating, it would actually produce an induced polarization in the molecule, and this polarization is directly proportional to the electric field, and the proportionality constant is the polarizability. Now, when I say polarizability, it is the polarized because it's only the electrons which will move because it is lighter compared to the nucleus, which is very, very heavy. So the electrons would move, and how easily they can be polarized in the x direction, y direction, and z direction is what this particular function is. And this means now the induced dipole will be oscillating with the same frequency as the excitation. And that means, and in 12th standard, you have been told any oscillating dipole will produce electromagnetic radiation, and it will be in all directions. And this is nothing but elastic scattering of light. Now, this is provided alpha is a constant. But alpha cannot be a constant because when you suddenly execute vibrations, when you actually move the atoms, then what happens is that electrons have to move with it, because in a vibrating nucleus, vibrating atom is also neutral, charge neutral. But at that point, when you actually put the electric field, this electric field will change the polarization, and the polarization will be dependent on the vibrations itself. And now vibrations are also oscillations, so it has a frequency associated with it. But these vibrations, because the bonds are very, very stiff, they cannot be large vibrations. They will be very small vibrations. And in mathematics, there is a formula which has been taught to you called the Taylor series, and the Taylor series is actually the expansion for this alpha. So alpha is alpha naught plus dou alpha by dou q, that is partial differential, at q is equal to equilibrium times q, and plus the higher orders of it, dou 2 alpha by dou q square times q square at q is equal to 0 is the second order of it, and third order. Because I am not going to consider the higher orders because q is very small and e is not very large, so I would actually terminate the series here. But in principle, if my E was very, very large, then I can actually see this effect also. And that is called the second order Raman and the third order Raman. But I will concentrate on the first order Raman only. Now, if I plug it back, I see suddenly, if I do the mathematics correctly, I have the dipole oscillating with this frequency and a dipole oscillating with a product of cos and cos into cos. And all of you in trigonometry would have been taught cos A into cos B is equal to cos A plus B or plus cos A minus B. So that's why I expand this. I will find now this oscillating dipole is actually oscillating with frequency which is the same as the excitation frequency. This is the elastic scattering. And suddenly I have two factors, omega L minus omega M. This is one factor, this is red shifted, and this is omega L plus omega M, which is called the blue shifted light. So suddenly one vibration creates two new frequencies, and similarly any other vibration would be producing many more such two frequencies. One is called the anti-Stokes Raman, which is blue shifted, and Stokes Raman, which is the red shifted. Now, this will be Raman active if dou alpha by dou q is not 0, and you know that differential of a function is the slope of the function, and slope happens to be 0 whenever it is maxima or a minima. So if it happens to be a maxima or a minima, that means the alpha which is changing, the function is changing like this in a parabolic fashion. At equilibrium, the slope will be 0. So alpha has to be monotonically increasing function, only then it will be a Raman active mode. So this is the selection rule for Raman activity also. So this is easy. I never used any quantum mechanics. So I can always show you how the diatomic molecule works. If you have a same atoms like nitrogen, N2, or oxygen, O2, H2, then if it is oscillating, when you compress the atoms, the electron cloud is coming closer, and when the electron cloud comes closer, it becomes difficult to polarize it. So it will be low alpha, and as you expand it, then the polarizability becomes more, and so the alpha will be larger. So it is a monotonic increasing function. So a slope at this point is non-zero, and hence it is Raman active. And if you look at this atom, diatomic molecule with HCl kind of a thing, HF kind of a thing, where one atom is different from the other, still when you compress it, alpha will be small and alpha will be large when it is expanded. So when you plot the alpha at the slope, it is non-zero, and hence it is Raman active. So in both the diatomic molecules, the vibration is Raman active. Now, just to show you that it also has an effect when you look at it differently. Now, if you look at the carbon dioxide, when it compresses, it will be low alpha, and when it is expanded, alpha will be large, so it will be Raman active. This is symmetric stretching. When you have an asymmetric stretching, what happens is that alpha will be dependent on one side. It is compressed, it will be less alpha, more alpha here, and the reverse would be happening in the other side. And when it's expanded and not expanded, it is in an equilibrium position, it will be the lowest alpha. So in that sense, it will be showing a parabola. And now if you draw a slope, it is parallel to the x-axis. That means slope is 0, and hence it is not Raman active. And this can be shown for the bending modes also. If you bend the mode also, it will be having the same effect. So that means in carbon dioxide, only the stretching mode is Raman active, whereas both asymmetric stretching as well as bending will be not Raman active. If you take a water molecule, you can see in all the cases, the alpha will be having non-zero slope at the equilibrium. So that means all of these water molecule modes are actually Raman active. This is the symmetric stretching, this is the asymmetric, this is symmetric bending, this is the asymmetric stretching. Now, the problem is that you would think that it doesn't matter whether I look at the Stokes Raman or anti-Stokes Raman, I would have the intensity same, and intensity is this prefactor. This prefactor is the intensity, and it happens to be the same. So you would think that I can do either the Stokes Raman or the anti-Stokes Raman. Now, what you actually find out is the Stokes Raman is this, and anti-Stokes Raman is this. This is the blue shifted light and the red shifted light. And you can see clearly the intensity drops down, and it drops down substantially as you go away from the elastic scattering. So it is not conducive to do anti-Stokes, and hence most of us would be doing Raman in the Stokes region. And this is because of the quantum mechanics. And quantum mechanics tells you the population of excited state and the ground state is different, and that's the reason. And here, I don't want you to see the dou alpha by dou q term, which is written in this complicated Hamiltonian expression, which is useful for people who are in chemistry or physics. But as a biologist, you don't need to realize that. What actually happens is that when the light is excited, the light actually excites the electrons into excited state, and it cannot stay there for a long time. It comes back instantaneously, giving out light, which is the elastic scattering. But because there are also vibrational states present, the ground state of the vibration and excited states, which is much, much lower compared to the electronic states, the electron has to follow whatever the vibration does. So there is an electron vibrational coupling which happens, and hence it can give energy to the vibration, and it can take it from the ground state to excited state. And when it now comes out, it is red shifted, and that's the red shifted light and Stokes Raman. And the reverse can also happen, that an excited vibration can come down to the ground state, and that energy could be given to the electron. And when it comes down, it is blue shifted. Now, all of us know that the population of the excited state is much less compared to the ground state. So this process will be more compared to this process, and hence this the Stokes Raman is more intense compared to the anti-Stokes Raman. So this is what is the reason why we do Raman in the anti-Stokes. Otherwise, everything else can be explained in a simple non-quantum mechanic principle. Now, whenever we record the Raman, we are always recording the Raman in the shift. What you usually do is, we are looking at the shifted frequency. So Raman is always written as Raman shift, not as Raman or centimeter of frequency. It is always written as Raman shift. So 0 is the elastic scattering. How much did it shift from the 0? And that is what is given as the Raman, that is the energy of the vibration. So whenever we see shifted frequency, the light which is coming, if you disperse it, it will give you certain colors of the light, and there will be intensity associated with it, and those intensities is reflected here. Now, in comparison, there is another vibrational spectroscopy which is called infrared absorption, and the infrared absorption also gives you vibrational results, but it is also not completely telling all the vibrations. So always vibration spectroscopy includes both Raman and IR together to understand the complete vibrations of the system. But every peak which you get is related to certain vibrations, and in this case these peaks are actually due to carbon tetrachloride, and all of you know carbon tetrachloride is a tetrahedral molecule, and tetrahedral molecules can only oscillate in this fashion if they are AB4 type of a molecule. This is asymmetric stretching, this is symmetric stretching, this is asymmetric bending, and this is symmetric bending. You can see here it is like a scissor motion. This is compressing, this is compressing, this is expanding, this is expanding. So it is perpendicular. Any tetrahedral molecule can be considered as two V's perpendicular to each other. And it is not only this. If I had CH4, then also the spectra would look the same. SO4, which is also a tetrahedral molecule, would look the same. So in fact, the spectra will look the same irrespective of which molecule it is, but the value of this would be determining whether it is CH4 or it is CCl4 or it is your SO4. So in fact, this is a very characteristic peak of symmetric stretching, which is around 415 centimeter inverse, which is actually the symmetric stretching mode of the carbon tetrachloride. If you look at the SO4, it will be at the same peak will be around 890. So you can see methane, it will be around 3,300 or something, 3,000 region it will be. So you can see that is the reason why Raman is a fingerprint of the molecule. So if it has the same configuration, the spectra will look similar. So isostructural systems will have the same spectra, and this is why it becomes very, very useful in fingerprinting. In fact, many of you would have seen Raman even can be used in a complicated molecule like DNA. So DNA fingerprinting can be done using Raman because of this reason that they have very unique characteristics when all the molecules look same. So that is why it is a fingerprinting technique also for molecules. Now, whenever you have a peak which doesn't look like a single value, always, whether it is NMR or mass spec, or you are having your spectroscopic, any spectroscopic tool, whenever you think that you have a peak which is more than 1, then you have to lower the temperature, because whenever you lower the temperature, the peak in width actually reduces substantially, and the peak reduces, and that is what happens in your system, and that you will find that many peaks. Now, what is the reason why this should have been a single peak? We have multiple peaks, and whenever I have asked, people have not been able to answer, but I have already given you the theory of Raman. Basically, the peak is because of the Raman, and Raman peak depends on two things. One is spring constant, and the other one is mass. Now, you know that in a carbon tetrachloride, your spring constant cannot change, but you have something called isotopes, and in that case, you have a change in mass. So you have two types of isotopes of fluorine, that is Cl35 and Cl37, and their relative abundance is 3:1, which means 75% of the chlorine atoms will be your Cl35, and 25% would be your you are going to have the Cl37, and 25% means 1 in 4. Every time I pick randomly a chlorine atom, after the fourth atom will be Cl37. So that means it is not a small ratio. Now people would ask, what about carbon? You know that carbon, the relative abundance is quite different. C12 is more than 99.9% or even 99%, and it's only 1%. That means I have to go 100 atoms before I can even pick up a C13, and it is still less when it comes to C14 itself. And so that is the reason. And another problem with C14 is also your C13 is a radioactive material also. In a sense, C14 is a radioactive material. I'm sorry. So now if you look at it, these peaks have meaning. This is the isotope effect. So all 4 atoms are Cl35, which will be lighter. And because it is lighter and in the denominator, this will be higher frequency. And next will be 3:1. This will be 3:2. And then you can understand that these two are 1:3, and all 4 being Cl37. So this is why the intensities are also affected by it, because the probability of finding this is much less compared with the probability of this. And you can see the relative abundance of 3:1 is the most tallest peak. That is it. Now from this, in biology, it is very, very easy, because if I do deuterate my sample, that means labile hydrogen, if I actually change by deuterium, it is a heavier atom, so my peaks in Raman should shift to a lower frequency. So this is very useful when I am looking at amide bands and many other things. Very, very useful techniques. Similarly, I could actually substitute with carbon or oxygen or nitrogen with a specific isotope, and I can actually, in a complicated molecule, look for frequencies from a particular region. So isotope effect is a very important thing. Now I will actually quickly go into the biology part of it. All of us know that we do end up with heart attack, or there is a block in your arteries. This is what is a very important thing, and many a times we want to know exactly the origin of it, or what is the level of plaque, or can we actually find out exactly where the clot is, because most of the times we are doing a MRI imaging or many other imaging techniques which we use. It doesn't exactly tell you the molecular nature of this plaque which is being built up. But Raman, I told you, is a fingerprint. So if I had this is a in vitro study. So you can see I have taken an artery which is normal, and if I take my normal artery, it is full of collagen and then elastin and actin. So my Raman spectra will be having those signatures. But when you have lipid-rich plaque, then it will be full of cholesterol, keratin, and proteins, and you can immediately see there is a change happening in this system, and your spectra is different. And now you also have calcified plaque, where calcium hydroxyperite is one of the major thing, and this is the peak because of calcium hydroxyperite. And you can actually see there is a huge difference in the chemical occurrences, and you can find out. Now, why did I show you this? Is because in real diagnostics, you know that we can actually put catheters inside, and you can actually make that catheter into a Raman probe. And or you do endoscopy, you have always a fiber attached to the end, and you actually look at things. So you can actually go to different regions and see whether the plaque formation is happening, what is the nature of the plaque, and what will be the effect of it when you put a stent, and all of these things can be done in situ. So currently there is a whole lot of research happening in trying to make miniaturized Raman and try to do these effects. Now, another very important thing is breast cancer. So one of the problems with breast cancer is that generally you do a complete removal of the breast when the cancer has developed in a larger extent. And this is actually a problem for the aesthetics of it, as well as you don't want to remove the good tissues away. So is it possible to actually find out whether the exact extent of the cancer? Because the laser can be pointed to about a micron size, you can actually take Raman spectra of each of the tissues, and when we do the tissue and average it, so actually we find out from our normal data, you actually create a fit for the Raman, and this is the live data. That means as the surgery is happening, you are recording the Raman of that tissue, and if it is a normal tissue, it will have this form, and if I subtract the fit from the data, I should get more or less no changes. So this means this tissue is really the normal tissue. And if it is a malignant tissue, immediately I have a change in my Raman spectra. And if I can actually find out this, immediately I will know the extent of the tumor, and I can remove exactly, a surgeon can remove exactly the right amount of tissues and leave the rest without disturbing. And you know that sometimes the breast cancer doesn't spread too much, so you could actually control it very easily by not removing the complete breast. And this is one example. You have skin cancer, you have oral cancer because of tobacco chewing, various other things. If we can actually do this, this becomes a very, very important tool rather than removing the complete tissues and disfiguring a person. So this is something which can be done very easily. And by the way, if you can detect it at very, very early stage, because the Raman can actually detect at epithelial level, so even before the tissue is building up, you can actually see the occurrence of tumor, and that's why this becomes useful. Now, another important thing which people can use it is that there is a Raman can distinguish between occurrence of DNA and collagen, actin, glycogen, many other important things which is present in your tissues. Now, this region which I am looking at is the functional region, and you can see there is a clear differences which a trained computer can actually find out. Now, whenever you do a staining of your tissues and see that this is a urinary bladder, and you can see two different types of your epithelial cells. One is lamina propria and urethrium. So urethrium is seen in the darker shade, and lighter shade is actually the LP. Now, if I do the same thing using this technique, you can see clearly the extent of the two, and you can distinguish between both of these things. So this is a very useful probe for tissue imaging itself. So you can actually image different tissues because the origin of various, these both are epithelial cells, but it has a distinction between both of them, and you can actually make it staining very easily. So another important thing is pharmacology. So pharmacology, you want to know whether your consistency is same or is it the drug is fake, because many of them make fake drugs. So you want to know whether it is actually made up of the fake drug. Here is an example of a paracetamol. Paracetamol which you get in the in drug stores. You see that paracetamol is actually having aspirin, paracetamol in a particular composition, and it also adds caffeine into it and cellulose, and there is a particular ratio a particular company has. And now if you do the imaging, you can see the red one is aspirin, green one is the paracetamol, and caffeine is the blue, and the concentrations also you can see that the blue is much less compared to the green and red, and you can see that the ratio of the red to blue is a particular ratio. If you really zoom in, you can start seeing the yellow region, which is the binder. Cellulose is the binder, and you can see the cellulose present inside that. And this is what is the binder which actually holds the whole drug together and solubilizes. Now, these are the different applications which I have just shown. This is a limited application, but we can also do many more things. But whenever I told Raman, I never told you that Raman is a very, very weak phenomenon. So if my concentration goes down substantially, suppose let us say I am talking about therapeutic proteins or any of those things. It is very difficult to do Raman of those things. So in that sense, one new discovery which is serendipitously happened in 1974, but it became much more better when the nano technology came in, is when you have a nanoparticles of gold or silver, and when you put your molecules there and put the light, that enhances the Raman signals. And this is called the surface enhanced Raman spectroscopy. But what is the reason why it happens? So whenever you have a nanoparticle, it is charged neutral, but it is made up of two charges. It is made up of nucleus and electrons. And you know that in electrons are, many of them are in the conducting electrons in a metal, and hence the charges are actually freely moving around, and so these two are present and they are actually overlapping each other, so it is charge neutral. But whenever you put a disturbing thing, that means it can be disturbed, the electrons will move and it will oscillate, it will start oscillating. And that one such thing is the electromagnetic radiation, because electromagnetic radiation has electric field and it can oscillate. Now when electrons oscillate, it produces dipole. All of a sudden you have positive and negative charge separated temporarily, and they oscillate and it produces electromagnetic radiation. So these nanoparticles start producing an electromagnetic. But does it happen for everything else? No, because you can see the colors are actually dependent on the size of the nanoparticles. So there is something associated with the oscillation of these dipoles. The color is because of that electromagnetic radiations which are actually absorbed or transmitted. This is what you all have studied, all of played with a swing. You all know that the swing has a particular frequency. If you push it, your swinging is of the arm is different from the swing's frequency, it will hit you. It will never go up. When you swing with the exact the frequency, you see the swing actually going all the way up. This is momentum transfer. That is called resonance. Resonance is that. So whenever the frequency matches with the frequency of the oscillator, you will actually have complete absorption of energy. And here also, this has a frequency associated with it. And if suppose you have the frequency match, it will get absorbed. If it is faster, it gets transmitted. If it is slower, it gets reflected. So that is the reason the same liquid which has gold nanoparticles. So this is the bigger gold nanoparticle. This is the wine red, which is a commonly used thing in gold, is the small nanoparticle. And if you see in transmission, it looks wine red, whereas in reflection, it looks brownish. Similarly here, so the colorations are different because of this reason of resonance. So you can look at the particle and tell whether it is a bigger nanoparticle or smaller nanoparticle. This is what you would do a technique called UV visible, and that is what you actually do for studying these colors. But it can be seen and talked about directly. So this is the surface enhanced Raman, and this can enhance the Raman signal by around 10 to the power of 4 to 10 to the power of 8, and hence it becomes easier for us to do Raman of single molecular nature. So this becomes competing for fluorescence confocal imaging part. Here is one quick example of my latest paper, which is to look at extracellular. Since the time is short and I want people to ask questions if there are any, I will quickly go through it, not to describe because most of you are biologists. I am actually working on extracellular vesicles which are under autophagic conditions. And this is because if these extracellular vesicles have distinctions in autophagic conditions where if it is cancerous or any other changes happening in your body because of a disease, these extracellular vesicles present in your blood and urine can be actually isolated, and you can actually see it as a marker for disease using Raman. So basically extracellular vesicles are nothing but membranes which is covered by a lipid membrane, and it has many things like proteins, nucleic acid, messenger RNA, microRNAs, and soluble proteins. But if they have CD63, CD9, and CD81, then it becomes exosomes. Otherwise, they are just extracellular vesicles. Okay. So now we actually use, because they will have similarity in EV, we use something called the principal component analysis, which is similar to what you do in your flow cytometries and other kind of things, where we use this analysis to look at sorting of the Raman data. Now, you can see that different cell lines we have used. This is the cancerous cell line, this is the non-cancerous cell line, and this is the ATG5, which is a very important autophagy related protein. We have knocked it out, and that is the cell line in HeLa which we have taken, and we have seen in DLS, that is dynamic light scattering, you can know the size of the exosomes. So you can actually find out, and you can also do a TEM imaging. And here is the nanoparticles being put and the surrounding the particles. So basically, we surround the exosomes, which are much larger, around 50 to 150 nanometers, and these are about 30 to 50 nanometers, and they would actually surround it, and we can actually get information about all of these things in the Raman. Now here is a Raman of three different cell lines, and this is the total exosome isolation reagent, which actually gives you the similar spectrum. By the way, whenever I have shown you this is actually done on 15 different samples, and each of these data is actually collection of 15 different data, and the average is given in this line, whereas this is showing that the repeatability is very, very high. You can see subtle variations. I will actually use them to do it. So basically, this is your the exosome extraction reagent, which is majorly seen. But along with that, you can actually do a subtraction of the two from each other, and you can see the differences are very clear from the Raman. So the differences you can actually see exist between different cell lines. So these are the points where the differences are happening, and we are actually going to look at these points and do a PC1 to PC2. And whenever you look at the cluster, we can draw an ellipse around it using an ellipse equation, and we can cluster them. Now this is how it is. This is the HeLa cells, and to compare it with the tier, that is the exosome extraction liquid, you can see they are much more tighter clustered, and here is your autophagy knocked out protein knocked out system, and this is your normal cell. So there is a clear distinction between each of them, and now you can even prove that if I get an unknown sample, so we were supplied an unknown sample, and we found that both of them were clustering around the HeLa cell, and this was only a quick blind test which we did, and then later on we found out that the supplied exosomes were from HeLa itself, and so we could actually distinguish. So what I have shown you quickly is that we can in fact distinguish a cellular component, and we can actually do a very quick analysis of that. For this, we need to actually develop benchtop Raman spectrometers, and this is something we have developed for ICER Trivandrum for doing pesticide detection. You can see that it's a spectrometer, and even if you can have a 96 well plate, this we have developed again, and we can actually do a 96 well plate format, this whole Raman spectrometer, which costs less than about 10 to 15 lakhs, and you can actually see the data. Now the same experiment has been done with the commercial Raman, which cost about 1 crore and above, and this one is the Raman spectra, and you can see the only difference is that there are fine structures which are lost because of the width of the peak, because the resolution is poor. You can see this is another Raman spectra, and you can see more or less reproduction of the most of the intense peaks without any problem. And you can also see it takes only 5 seconds to do this with the same milliwatt of the laser, and say this is about 4 to 4.5 minutes that you require to get the same intensity. So it is actually possible to do these things. Now, I will stop here. I've exactly finished about 45 to 50 minutes, and I will be open to questions from your side. But I hope I have actually shown you that Raman has a very large potential in doing in biology a lot of stuff. And this wouldn't have been possible without all my students who are here. And thank you very much for your attention. Santosh, I have done my talk, and now people can ask questions.
Okay, thank you, sir. Actually, now basically, I just before that, I would like to express sorry. There is actually, sir, we basically this meeting will allow only 250. Actually, 330 itself, we have exceeded this number. So we have to block the remaining participants. I'm extremely sorry. So many probably will be waiting to join. Probably
but it is recorded, so that can be shared with them. I think it is recorded. So I think you can actually put it on a place where they can actually go back and listen to the talk. If everything was clear, then they would be able to do it, and they could always contact me over email or in person and ask me questions for those who couldn't attend. I'm really sorry that this platform doesn't support more than 250.
Okay, now it's open actually. Questions.
I will stop sharing so that people can see each other.
Sir, actually, I have one question actually. When tissue is actually imaged for plaque identification characterization, basically the tissue we expect a lot of complex biomolecules actually. So this E spectra, I assume it is going to represent unique kind of molecules. Then being biology or surface being much more complex, we expect that the spectra is also going to be much more complex actually. Do you see such situations actually? When
absolutely, what you said is absolutely correct. So basically what happens is that the Raman spectra of every organelle is different, and if I actually have a laser spot which is much bigger than the size of the cell, you know that cell itself is about close to about microns size, a few microns. If my laser is bigger than that, so it will be actually a convolution of all the things which is present. So my Raman spectra will be a complicated spectra. Yes, absolutely right. Now here is where the modification is done. Now what we do is that there are many ways to target a particular organelle or target something. Like if you want to target a mitochondria, if you want to target something in a cytoplasm something, or you want to actually look at it, then what we do is that we either do a near field Raman. Near field Raman means you actually focus your light much sharper than few nanometers, few hundred nanometers. So you can actually go to nanometer level your focus. So now you start separating out organelles. This is called near field Raman, where this is near field is a good technique to use it. Another one is TERS, tip enhanced Raman. So there is something called you use an AFM and you go over the regions, and wherever the AFM is actually having gold nanoparticles, you would have signals coming more. So this is called the TERS setup. And the other thing is you also do something. If you are a poor man's, so we don't have all this technology, then it is very easy because there are specific signatures for certain molecules. So those regions are actually identified, and you put a window around it, and you compare the intensities of them, and you then map it. So this is what is the technique, like what I had done in the case of pharmacology experiment, where I had all these samples are present at the same time. Now that is aspirin, your paracetamol, caffeine, and cellulose. All of them are present. So my spectra will be having all of them. But at any given point of time, I would be getting a spectra more from caffeine or more from a particular region, because that happens to be more concentrated. So there I would get more signal. So I will plot the intensity of those and then shade it around. So that actually makes it easy for me to find out this. So these are many techniques available now. So these are the techniques used for tissue imaging. But everything you have to train your system. And once it is trained, and AI can do it. Artificial intelligence can do that, because it immediately learns that for this tissue, these are the signatures for, say, a particular type of cell, and then it gives you an information about it. So this is possible. And you can do confocal. You can do confocal, because you can actually go from one to another. Yeah, go ahead.
So that means that beam shaping, you can even increase the resolution through laser beam shaping.
Yes. So that's what I said. Near field is one this thing. Most of the times the spectrometers comes in the far field geometry, but you can go for a near field geometry, and that will do the beam shaping very easily. Yeah.
I have a very basic question, sir. So since the Raman spectra is given by vibration of the bond, so in proteins, I understand that is because of the peptide bond, and then side chain that will give. But when you say about the Raman spectra of DNA or RNA molecule, all bonds are same. So will the length of the length of the DNA or RNA will give a different spectra, or it will be just like UV, which gives the same spectra for irrespective of the length? But I can understand that it can differentiate between a single strand and double strand, because their bonds will be different. But will it differentiate the length?
So very good question. Very good question. It is not the length we are looking at, because when you look at the DNA in toto, it will be much larger than what your laser spot itself is. So we actually are looking at the nucleotides. We are actually looking at nucleotides. And in fact, people have tried to use this to distinguish ATGC sequencing also. This is a slightly more being developed now. So you can actually look at the ATGC combinations, and you can actually say whether the sequence is the same. So it can, at that level, distinguish between a DNA from one to another. So if I don't have that sophistication, I still can distinguish between a bDNA and a DNA, or all of these things, because I told you there are certain configurations that is there in a molecule, and it has a particular signature. This signature is unique, and that cannot be repeated until unless it is the same species. So that is the reason you can even distinguish DNAs, you can distinguish proteins, you can distinguish. So if I have a lysosome, or I have other types of proteins like say HAT, histone acetyltransferase, or something else, all of these things have a different signature. And this is very, very unique. And even in a crowded situation, I can actually get this easily identified. So that's why called a fingerprinting technique.
Okay. So here another question I have, when you mention about this, you can nail it down, is because when I think about this vibration, I always think about the effect of solvent, what contains that might affect the vibration of the bond. So when you look at this complex solution, then how do you subtract or negate the effect of the solvent?
You are saying solvent, right? Solvent.
Yeah.
Solvent. Most of the time, that's what I showed you in my exosome. That is basically the EVs, extracellular vesicles. I had a huge contribution from the solvent. There was a huge contribution from the solvent.
What
So what I
mean
do
is,
is I actually repeat the experiment multiple times to see whether the changes which is happening in the spectra is repeatable and highly consistent. And if they are, then I average them and then do a subtraction of the spectra. So when I do the subtraction, those subtle changes are repeated every time. And these
okay,
subtle changes is what is the signature of the presence of that.
okay,
Ideally, I don't like to have the solvent at all. So many a
okay.
times if I use phosphate buffer or I
Uh-huh.
use those kinds of things, the
Uh,
signatures are very weak in my
uh,
Raman. Phosphate
my
buffer.
question was this: will solvent affect the spectra of that? I'm not talking about the spectra
Oh,
of the
oh,
solvent, but I'm
okay.
talking
Okay, I
about if
understand.
what I
So
mean
in,
if you
in
change the
principle,
buffer.
they do not
Uh,
affect the spectra. They
okay,
are convolution of the two. That means
okay,
solvent spectra plus the sample spectra. They never interfere with each other until unless they form a bond. They form
okay.
something. So that means they should have, if suppose let us say I add
Uh,
methanol in water. If I
uh,
add methanol in water, then methanol and water actually forms hydrogen bonding.
yeah.
And because of that, the methanol peaks will shift, but methanol remains methanol. But
Okay,
there will be a shift in the peak, but not
okay,
really changing the methanol Raman spectrum.
okay,
So
okay,
there
okay.
can be a shifts which will be caused because of the interactions, but it will never be changing the spectrum.
Change
So it is
uniqueness.
very unique to that region. Yes, I got it.
Thank you.
Hope I explained that. Yeah,
Yeah, yeah. Listen.
That
Yeah, hello. Uh,
yeah. Go
uh,
ahead.
yeah, that was a really interesting talk. And the
Thank
question
you.
that I have is regarding the breast cancer tissue that we are talking about.
Right.
Uh, so, in a breast cancer tissue, you have, suppose you have the cancer cell and the normal cell adjacent to each other. Like, what would be the resolution of differentiating between these two? Like,
So in general, it is in few microns. The resolution, spatial resolution is in microns. So
okay,
about 1 to 2 microns is the spread of the laser. So in fact, when you are actually looking at it, it will be few microns separation that you can actually do it. So you can have the imaging setup, which can actually be moved within a precision of about, I mean, you can move it in nanometer scale with using a piezo translator, but you can actually have a resolution up to about a micron in your
okay.
imaging. So if I do an imaging, I can get a distinction within a micron resolution in space.
Okay, so do you think like this could be also applicable to understand the, I mean, the brain neural stem cell niche, for example, because the stem cell is in a niche and you have surrounding neurons along with it. So,
Yes. So actually, these are the reasons why I actually moved in here, so that we can try these experiments and see it. Because all of these are in the, see, biologists have never used Raman very
exactly.
much because they have been always happy with the instruments which they have got. But now it opens up a possibility. But never ever a complete biologist can do this. So you need an interdisciplinary approach to this. So we will have to collaborate and do it. So yes, in a zeroth order, I can say I can actually distinguish. But when we actually go into the setup, so that is what I am actually trying to set up here. So I will be doing it. So there are 2-3 things I didn't discuss in this is UV Raman. So UV resonant Raman is there where I can actually pick up tryptophan. I can pick up a particular molecule in a protein, or something can be enhanced by using UV Raman. So that is the way we can actually target an image and do things. So we can, if I'm looking for a protein, I can look at for a particular signature in that, in a peptide or anything. So these are things possible. So this is exactly the reason I think have a lot of things to do together scientifically. So I will be interested in taking up and doing on stem cells. In fact, Manisha Inamdar, you know that my colleague
Right.
has been trying to work with me. Unfortunately, we couldn't go forward, but now I am sitting inside this. I'll be much more easily able to do those kinds of things. So Manisha has been working with me, and you know Manisha very well. And so we are interested in actually doing stem cell differentiation and other things. So we should be able to look at neurons and other aspects of it. Yes.
Thank you.
Sir, I have a question.
Your voice is not heard, Pradeep.
Hello. Sir, I have a question.
Okay.
So, this is with respect to the exosome you are showing. And my question is like how much volume it will be required actually to do the exosome analysis in
could you repeat that?
How much volume of exosome it will be required? Do you need a large amount of?
We didn't need too many. I mean, of course, it has to be sufficient enough inside so you can actually do look at it. If I have to have sufficient enough exosomes in a volume of 1 micron by 1 micron by 2 microns, that is the cube which I'm talking about. Inside that, at least a few exosomes should be there. So if I look at it, it would be the concentration you can quickly calculate. It would be somewhere around a micromolar or something like that. That should be good enough for me to actually work with exosomes. If they are in micromolar concentrations, that should be good enough. So we are trying to go lower, but micromolar is possible to get. So we have been able to extract exosomes and do that. Yes, go ahead.
So, sir, if the freezing and thawing, whether it will affect the exosome, the Raman scattering?
We need to do freezing and thawing. Yes, exosomes. This is one of the things which we were doing. We need to do freezing and thawing. That doesn't affect the exosomes at all. It remains intact, and Raman comes all right. Yes. Right now, we have gone into another cell lines, and there we don't need this extraction reagent. So if you can remove the extraction reagent, the data is much more better. So my student is actually working with other cell lines, which came from the hospital. We were working with the autophagy group. That's why they were using this extraction technique. But in a clinical setup, they can actually extract the exosomes much better without any of these interfering liquids. So right now we have data, so we are actually analyzing that. So we will have a much better understanding of that. Yes.
Thank you.
Pradeep, you were asking something. Not able to hear. Yeah, now it's better.
Okay, you can hear me now?
Yeah. Yeah, I can hear.
Okay, so the thing was, the question that I was asking was, so can we use Raman to probe the live cells? Can we look at the living cells using Raman?
Look at what? Look at what?
No, I cannot hear you. 1 second.
No, it's not coming. With the earphone, it is not coming. Your mic has got a problem.
Yeah, my question was whether we can image live cells using Raman.
Yeah. So basically, cell imaging is now one of the things people are actually interested in. So, Santosh mentioned, we have a lot of complicated, it's a convolution of all the spectra. So we will have to really train ourselves. So there is a requirement for not only just doing Raman, but also to train our computers to actually decipher these changes which is happening. And once an AI is included into this, it becomes a possibility. Now the AI is very easy to work with. People are actually looking at AI very significantly. So we could actually use the AI and the Raman spectra analysis together to actually do a cell image, live cell imaging. So this is something which is interesting. So one of the things we were actually interested in, can we actually look at in one cell at a time in a flow cytometry? So that is something which is we are trying to understand if one cell passes 1 second, can I actually distinguish whether this is can can I actually get better information than what the flow cytometry gives? So those are things people are actually right now working. Yes. Yeah, somebody was asking. Yeah, now go ahead.
Yeah.
Yeah, you asked.
Can you hear me?
Okay, yeah.
Yeah, this is
I
Malini.
think, yeah, Malini. Yeah, tell me.
Yeah. Hi. Yeah. So, I was wondering whether one could look, measure the depth, if you have a cell and you have a glycocalyx around it, can one measure the depth of that glycocalyx? Like in ESR, we can measure the membrane depth. But in Raman, is it possible?
Yes. So it is exactly like confocal. So in fact, we have confocality in Raman. So because of the confocality, you can slice the whole depth profile. In fact, you can go from one level to another level. The resolution is about 2 microns. So you have to have something below 2 microns to actually be seeing, because always laser light, in fact, goes into the system, and you will be collecting plus or minus 1 micron around it. So about 2 microns is your depth resolution. So if you are whatever you are looking for is bigger than the 2 microns, you can actually see it. If it is less than 2 microns, it will not be seen. It will be seen as a dot. It won't be seen. Much information will be there. That is the depth profiling. Exactly like your confocal. Fluorescent confocal imaging. Super resolution. Exactly like that.
I also had a comment on your that PCA figure where HeLa and all was. So the HeLa and your buffer were coming together.
No, actually, buffer is to show that it is many of the peaks. So many of the peaks are coexisting in these things. That's why they are closer. So very few subtle variations are there, and that subtle variations is making them to be separated out in space. So that is the reason. So in fact, now the new cell lines which we have got from the St. John's Hospital, we do not have to have this this liquid. That is the exosome extraction liquid is not necessary, and that makes our life easier because there won't be any interference from this. The differences are much more better to see it. Yeah, somebody was trying to ask. Yes, go ahead. Go ahead. You are raising your hand.
Sir, I had a question. Yeah, that was a very interesting talk. So we work on bacterial system. So my question was, can Raman be used to detect heterogeneous population? Like when you treat antibiotic, you'll have some dead cells, living cells. So can Raman system can be used to sort or analyze heterogeneous system?
So what exactly is the question? Why do you want to distinguish them? What is the reason you want to distinguish? Because the reason I'm asking this question is because because most of the bacterial systems can be imaged with a visible microscope or even by other techniques very easily. Now, if you want to do Raman, you should be asking a much more profound question, because otherwise people will say, why do you want to use another technique to do it? So the question is whether really you want to know the information inside, like molecular information. Molecular information about these things, does it make any important information? Then Raman is possible. Of course, it will distinguish a dead cell from live cell and all of those things, and different types of bacteria will have different proteins on it and various lipid configurations. So it will actually give you a different spectrum. Now, the question whenever we ask this is that, does this give better advantage than other techniques? That is the question we should ask. So if other techniques can do exactly like what Pradeep was saying, that I can actually get this information with other techniques, then why do you want to do Raman on that? Because it will be a whole lot of investment of time, and people will say nothing new you have got. So we need to actually ask questions where other things are not seen. So that is why I would say, if you ask a more valid question, yes, distinction can be done, but we need to actually see that this is not possible. In the case of exosomes, exactly the problem.
Because
Exosomes are very difficult to distinguish with many other techniques. So they are trying to find out an easier way to distinguish exosomes. That is because already people know it is having some disease manifestation possible. So if it becomes easier to do it and you can do it without too much of complication, then otherwise you can do mass spec. You can do many other things. You can actually put it in a mass spec and see what are the compositions which are present, right? But how difficult it is to do a mass spec. But here you can easily do it because PCA is much easier to do it. So that is the question we should ask, that this should be more advantageous to do it. So if you make it more better, then I can answer you in a more affirmative way. Hope I clarified your doubt.
Is the reason is the antibiotic, the survival population? Yeah, just to kind of follow it up.
It can distinguish. Yes.
Yeah, because the persistent population is quite different from the dead population. So there is some kind of internal changes which are not can be analyzed by specific marker. So that's the reason I was trying to understand it.
So in fact, I'm saying it is right now my attempts are for single nucleotide polymorphism. So you know that alleles, when you are looking at, I am actually looking for one nucleotide which is actually changing. So I use a PCR plus, limited PCR plus Raman to actually pinpoint the allele which is actually changing. So, so I can actually do this for biotechnology applications, like go to the farm and if I can get the extraction of the DNA. So if I can distinguish that, I should be easily able to distinguish this also, right? So these are possible. Yes, this is possible. Yeah. Om Kumar?
Yes. My question, yeah. Can you hear?
Yeah, now I can hear.
Yeah. Does Raman spectrum give a characteristic, spectrum for a protein? Can we get a characteristic spectrum for one protein?
Yes. So my work, major work for past 15 years have been on proteins itself. So every protein has a very distinct Raman spectrum. So again, I said it is a congregation. Protein is actually not few atoms like what I showed you. It is a complicated amino acid sequence. So the primary sequence, the secondary sequence, many other things happen. So we actually target the secondary conformations. So secondary structure. That is what we that is highly dependent on the amide bands. These amide bands, depending on alpha helix, beta sheet, or random coils and others, their populations are different in different proteins, and they actually can be easily quantified by normal Raman. So we can actually see some conformational changes. So we ask questions like, suppose a drug binds to a protein, does the changes, can you see a conformational changes in the system? Yes, we do see. And whether it is HAT or whether it is looking at other kinases, or we are looking at proteins related to some other therapeutic proteins, we can actually see changes and we can actually distinguish between different proteins also. So this is one protein can give a different spectra, totally different spectra, and it is like a fingerprint.
All right. So, but then when we have a protein in vitro,
yes,
versus when the protein is inside the cell, will the protein will it be distinguished? Will it be possible to distinguish the protein? Because inside the cell, it could be undergoing several different conformational changes or interactions.
Very, very good question. Because in see, right now, very difficult. I told you, Raman is a very weak phenomena. So if your concentration is very, very less, then Raman signals are very, very weak. So it is not that we cannot distinguish, it is because of the weakness of the Raman signal. So what do I do for most of my protein study? I add a nanoparticle. So if I can target my nanoparticle to go and attach itself to close to a protein, and then I can image that, or I can get information about the protein, then protein would give me sufficient enough intensity. So I should be able to get it. But it is still a needle in the haystack condition So at this point of time, Raman has not reached that I can actually get all the organelles in your cell. So I cannot actually look at a DNA inside a cell. I cannot look at the lipid in a cell and distinguish each of them right now. But this is where we are getting to. We are actually trying to increase the possibilities by taking the Raman to with many other techniques. So what we can do, if I want, see, there is an indirect method. That's what confocal always uses. Suppose you want a confocal to be done for a particular cell, what do? You actually add fluorophores, or you use a FRET technique or many other techniques to actually do the imaging. So it is an indirect method. So if this happens, it is present. So we can also do Raman in that way. We can add some molecules which can give you a very strong Raman signal, which gives you a Raman signal when it is next to it. It doesn't give you a Raman signal when it doesn't sit there. So this is the methodology which we can use, and this is something we are developing right now on a similar way as a confocal imaging happens. So indirect method, so not direct. So you know that if this protein is there, this will go and bind to that protein. If I add to this, this will go and bind to it, and it has a marker, and that marker gives me a very strong Raman signal.
Hello?
So I can actually say that yes, this is where that marker is there. So this is much more better than confocal, because confocal has two problems. Yeah.
Okay.
Yeah. You have a question?
Sure.
So can I ask one question?
What I'm saying is that fluorescence has a possibility of bleaching, photo bleaching. So it doesn't have so it has a more advantage of it. I think Santosh wanted to say something. Santosh? Yes? Santosh?
Sir, can I ask a question?
Yeah, please. Go ahead.
Sir, your slide on atherosclerosis and Raman spoke, that gave me a sort of an adrenaline rush. So I wanted to ask you, one major issue in cardiovascular diseases is that, how to determine whether a plaque is stable or unstable. That is, whether it will stay for some time, or will it, thrombose at a very odd, weird times and give you a myocardial infarction or a heart attack. So is it possible to use this Raman scope which can determine plaque instability? Like,
very good question. Actually, currently, what I have shown is in vitro. That means I have cut your artery and opened it up, and I have shown it. The major thing which we are developing right now, or people around the world are developing, like in Harvard Medical School and Johns Hopkins and other places where medical technology, or MIT, what is being developed is, can I actually use the same cathedra which actually goes inside into the put the stent? Can I actually use that as a fiber? So, and I have an optical setup which is actually 3D printed or something, and I can collect the signal from those places. So once this instrument is ready and it is approved by the FDA that it doesn't cause any problem to yourself or anybody, then this will be possible to answer that question. That because the molecular signature is very, very unique for whether it would dislodge itself or it will be stuck there. Because after few studies, you will easily be able to find out the signature, because already we know the signatures of which would be dislodging, which will actually stay there, or what kind of plaque it is. That information, microscopic information, we can get. And we don't need to analyze the complete spectra. We need to actually do this by training our system. Like I showed you in the case of breast cancer. You train your system so that it actually looks at the spectra, and now you have a recorded spectra, and you actually superimpose the spectra you got. And if it happens to be matching 100% or 99, 95%, then you know that this is that type of a system. So this is what is the new development which is going to happen. It is still, being developed right now as I speak. People are actually interested in getting those miniaturized micro Raman probes where resolution will not matter, but it will be the signatures which will be mattering. So that is where we are actually working. So this is an ongoing process. But before that, we can do all the studies like post-mortem. Like take the ones where they got dislodged and get the signatures, make a library of it, then look for these things. Yeah.
Okay. Thank you.
Sir, I have a question. This is just about that breast cancer you were explaining. How effective would be the margins? You know, it is not like a single cell or like a blood vessel where the plaque is there. So this is like you have multiple cells. It is a tumor microenvironment, and there will be micro metastases, which are very smaller. I mean, cells which are very fewer in number. So how effective this Raman spectroscopy would be to identify the margins?
Yeah. So this one was done live by Harvard Medical School. The one which I have shown is done live by Harvard Medical School on a operated person. So that means the person was being operated and this was a live imaging. So this is actually something which people have done it. But yet this is not imaging. This was actually using a fiber. So the fiber is going and you are moving with physically. So at this point of time, there is no imaging possible. Now, the advantage of MRI or any of those contrasting things which are available is that you just take an image of the whole thing or find out the extent to which it actually happens, and you know how much is the growth of the tumor, your cancer. Raman has not reached that level yet. It has not reached. What it can do is, while you have already imaged by your MRI, and you have got an image, so the doctor has opened up that region and you are actually looking at it. You want to see whether leftover tissues are there. You are not interested in the whole tissue. You are only interested in whether there was any leftover tissues. And this leftover tissues can be actually looked for using Raman. That is where the problem comes. If you leave some tissues there, and that tissue will actually be causing the cancer in the patient later. That is the issue. So we have to complement the techniques. So you cannot exactly say that it will replace MRI or it will replace other techniques completely, whichever is being used. It will only complement the where the difficulty is there. While the surgeon is actually removing it, he or she will be able to see the extent of the tumor, because when you start removing, you start seeing that it could have actually proliferated. And main thing is, the cancer at epithelial level infections are never detected by any of the techniques. So this can actually look at the epithelial level. So at that level, you can actually get the signature. So these are advantages we have to buy. So you have to pitch it correctly. Where do you put your Raman? So if you say I have to image the full tumor, no, Raman is not the answer for it. But if you want to look at what is remaining, is it cancerous or not cancerous, then it is much more useful.
Thank you, sir.
Yeah, there is a question here. Yes, go ahead.
You can hear me?
Yes, I can hear you.
Yes, sir. I'm Nimi. So I'm not asking anything regarding the science. Like, as you are joining as a new director, as a student, what are the opportunities that we will be getting from, like, what we can expect, or like how the interaction opportunities that we will be having? So we would like to know.
Very, very, very good question. Actually, I didn't tell about that. So this is the right time to tell everybody. In fact, I am going to meet most of the faculty one-on-one, and that I am setting up the timetable for it. So that will take me some time for meeting the faculty itself. But I am planning to also interact with students. And since students are in larger numbers, so I will actually target the groups, or I will actually have to do an interaction in an environment which is safe, and we can actually have a conversation together. So I will be actually holding interactions where with people in the range of 30 to 40 people together in a lecture hall on one-on-one. Okay, so that means I will be physically present. You don't have to do this video type of interactions, which is, I find it not very effective. So physically you will be able to interact. This can be done. I will actually, so please do send in your requests for what interactions you want, so that I can be prepared, and when I come to you, I can actually talk about it directly. So that is what I would like to do. And this is not one time. This is not one time. Throughout my tenure, how long I am here, everybody can send in a request that this is what we want to discuss with you, and this is the reason I want to send it. My office will actually arrange for the interactions. If you have a genuine or alone a single problem, then you can come directly. If it is a collective problem, I can meet you collectively. So there is no need of any layers of intervention between. You are directly accessible. Is that a good answer? Yeah. This is for also for the faculty, and this is also for the technical staffs and everybody. So I am accessible to everybody directly. Only thing you have to tell me is whether you are meeting me in a group, whether you are meeting me alone, whether you are meeting me for a particular purpose, so that I am prepared. I don't come there and then look for, okay, I say, I have to find out the answer, I have to come back. If you tell me ahead of time, I will do all the background work and then come there, so that I can be more useful in that interaction. In a short time, I can solve the problem, hopefully. And main thing what I want is that you should be motivated enough. One of the things which students have a problem is they get depressed. And I think that is one thing which should never come in your student life. Depression is a part and parcel of research or study, but that should not remain forever. It should be removed from your body immediately. And for that, you need to open up and talk to people who you are confident with. So that's why these mechanisms are done. And we will have many mechanisms depending on how the problems are panning out. We will actually create panels. All these faculty members, in fact, I say whatever Professor MRP had done till now will not be anything changed. It will only be enhanced with all of your contributions. Is that okay? Is that a good answer?
Thank you, sir. Yes. Thank you, sir. That is wonderful section. Thank you.
Welcome. My job is only to interact with all of you. I mean, I have to do 20% of the time only research, but other things I have to take care of all of you. So you can imagine my research will be hampered, but this is the job. Yeah. Santosh, I don't think any more questions are there.
That was okay. If there is no
a small doubt.
Yeah, go ahead.
A more technical one. So at the start, you said that there are different types of Raman spectra. Like there's vibrational, there's rotational, and you focus on vibration. Is there a reason for that? Does rotational not give you as much information as vibrational, and there was one more type?
Okay. So rotation actually is because it is basically the molecules are rotating, so there is no much variations happen because of the environment. And when can something rotate? They can rotate if you are gaseous. You don't rotate if you are, you cannot have a rotational degree of freedom if you are bound by many directions with bonds. So rotational information will be more useful in gaseous systems. And naturally in biology, we are not talking about gaseous systems. Okay. So that is why I said there is a limited information which you can get. But when we look at the gaseous system, suppose let us say I am actually using it for interstellar studies, or I am looking as a Mars probe, then the gaseous systems actually matters for me. So there the application is useful. Rotational Raman becomes a very useful tool. Then electronic Raman, generally in biology it will not be useful for you, because in biology electronic Ramans are not very important, but in chemistry, where you have an excitation happening between an electronic level and how that excitation, so let us say pi star interactions. if you know some about chemistry, you know that there is all the UV visible, you have a pi pi star, n star, or sigma sigma star transitions. These transitions will be actually showing up in your Raman as a excitation, and those kinds of excitations give you a little information. So you can actually use them for understanding those kinds of interactions. So in fact, there is a limited effect of rotation and electronic. Others are mostly vibration. Raman gives you more information for biology. That's why I stuck to biology, this vibration Raman.
Okay, sir. Thank you.
Hi, sir. I have a question.
Yeah, go ahead.
Sorry, your talk is very informative, and I would like to ask you one question: whether exosomes they will have any background issues when you do the Raman spectra? So something like exosomes, like we have a lot of heterogeneous exosomes. I mean, in a cell. So does it give any background?
So yes, so we didn't do exosomes in a normal environment. We have actually extracted the exosomes. So we have tried to concentrate the exosomes from the cellular matrix. So we have isolated them, and we have actually tried to minimize as much of interference as possible. So we are not imaging the exosomes. We are actually trying to detect the exosomes. So we have extracted them. So in that sense, there is no interference. But again, my spectra had an interference because I used a exosome extraction reagent, and that reagent was leaving some mark in my spectra. That is why I had a tough time. But now there are methods to extract exosomes without all those complications. So if you can
traditional way, like
we can actually detect it without any problem. Yeah.
Okay. Thank you, sir.
Thank you.
So, if there is no more question, shall we conclude, sir?
Yes. Thank you very much for this opportunity. I got a chance to talk to everybody, and I would like to say before you conclude that all of you, please be free and interact with me. My office will be always facilitating your interactions with me. And I am going to visit your labs also. I am also going to visit various people and also facilities. So I would be getting a chance to much more closely interact with you. So I think you should feel free to interact with me. Thank you.
Thank you, sir. This was a very interesting, exciting, interactive section. Basically, it was so great to hear from you the most basic aspect of Raman confocal as well as its advanced application in almost all the areas basically where we are working. Actually, that is the more beauty of that. So it looks like everybody is sensitized with this system. Probably you are going to get more and more interaction with you by all the faculties as well as students in the scientific domain actually. And again, I also would like to express all those faculties as well as students who joined for this talk, as well as interacting with them. And again, I am, expressing sorry as well to most of the faculties who were not able to join. Actually, I got several messages and emails that they are waiting to join, but unfortunately, we will have to block. As you said, we will make arrangements, probably if possible, one more talk in a different occasion. Thank you, sir. Thank you all.
Thank you so much, and have a nice day. Thank you very much.